Hello everybody. In this lab we will 1. Learn about reactor hydraulics, and 2. Measure the hydraulic characteristics of a plug flow reactor and a continuous flow stirred tank reactor using tracer tests, and compare the results of these tests to the ideal plug flow reactor and continuous flow stirred tank reactor models. The possible flow patterns in continuous flow reactors are bound by two extremes: plug flow and completely mixed flow. These extremes are represented by two reactor systems, the plug flow reactor, or PFR, and the continuous flow stirred tank reactor, the CSTR or CFSTR. The hydraulic regime, or flow pattern, within a reactor can be characterized by performing a tracer study with a conservative, meaning non-reactive material, and analyzing the resultant data using various hydraulic models. During the experimental portion of the investigation, a known amount of the tracer material is introduced into the reactor inlet, and its concentration in the reactor effluent is monitored over time. It is important that the flow rate remain constant and known throughout the tracer study. Please wear all required PPE while performing this lab: lab coat, gloves, safety goggles, and with any long hair tied back. The CSTR system is set up, and water is pumped into the beaker, the reactor, using a peristaltic pump at a fixed rate. The outlet of the reactor is where the samples will be taken. Calibrate the outlet flow rate to 200 milliliters per minute. Fill the beaker reactor. Stir the water moderately so as the stir bar does not jump at the bottom of the reactor, and open the outlet valve. Collect the water using a graduated cylinder and start the stopwatch to count the time. Based on the water collected and time, you can calculate the flow rate. Adjust the valve open position to adjust the flow rate. Once the flow rate is set at 200 milliliters per minute, keep the valve in that position. Once the flow rate out of the reactor is set, calibrate the pump to the CSTR so that the volume within the beaker stays at 1 liter. Start the pump and adjust the speed until the reactor volume is constant. Using a pipette, add 1 milliliter of 1000 milligram per liter methylene blue directly into the reactor and start the stopwatch such that the moment that methylene blue enters the water of the reactor is T equals T0. Start collecting the effluent into test tubes every 30 seconds for the first 3 minutes and once per minute thereafter. Record the absorbance of each sample at 652 nanometers. Continue the run for 10 minutes or until the effluent concentration is less than 0.2 milligrams per liter, whichever comes first. Measure and record a final flow rate. The plug flow reactor system is set up such that water is pumped into one end of the tubing, and the other end of the tubing is the outlet where the samples are taken. Calibrate the outlet flow rate to 200 milliliters per minute. Fill the reactor with water using the peristaltic pump such that there are no air bubbles in the reactor, and open the outlet valve to let the water run out of the reactor. Let the water run for a minute, then start collecting the water using a graduated cylinder and a stopwatch to watch the time. Based on the water collected and time, you can calculate the flow rate. Adjust the pump speed until you get 200 milliliters per minute. Measure 1 milliliter of 1000 milligram per liter methylene blue into the syringe and pierce the syringe through the septum of the T-shaped connector at the beginning of the tubing of the plug flow reactor. Allow the pump to run for a few seconds before injecting the methylene blue at time T equals T0. Do not remove the syringe. Begin taking samples once the slug has moved approximately 3/4 of the way through the reactor. While the slug is visible in the effluent, collect the effluent as frequently as possible into test tubes and record the time each sample was taken. Read the adsorbance for each sample at 652 nanometers. Continue the run for 10 minutes or until the effluent concentration is less than 0.2 milligrams per liter, whichever comes first. Measure and record the final flow rate. Be careful with test tubes, as a broken test tube may cut your finger. Be careful of water on the floor, walk slowly in the lab, and be mindful of the methylene blue dye, as it can stain your skin for a long time. After this lab, you should know how to analyze the flow models in plug flow reactors and continuous flow stirred tank reactors.